2. Very good. There we go. All right. Good. Sorry, I'm just trying to find my spot again. There we are, right there. Perfect. Okay, so just to continue, what we have here, we went through all the different and I'm not gonna ask you for, anything there about the plant. Okay, I just wanted to show you guys a comparative analysis. Okay, the complexity, and that the bacteria is not complex. As evolutionary, it's very simple, but it's very effective, and it's here forever. Okay, so we've got these guys here. We've gone through that, and there's our ribosome. Okay, here is our that's the bacterial DNA, and binary fission, division into 2 cells. So we've gone into conjugation. Conjugation allows for a form of sexual recombination as genetic information exchanged between bacteria. So that's conjugation. Transformation. Bacteria can undergo transformation when they incorporate DNA from the environment into their own genome. So that's transformation. Okay. Transduction. Okay. Transmitting viral DNA from one bacteria to another. And the spores. All right. And as I said, these spores can be around for thousands of years. And going back into the whole permafrost, the permafrost there in Canada's and Russia's north is thawing, which is now providing the environment there that these endospores can germinate. And you see there's not much to them there, okay? These endospores are highly resistant, and this is actually the test for an autoclave. These are the toughest ones. These things can handle high pressure, high heat. Okay, and yeah, these are this is the tough part here. So you look at the endospores there for the genus Bacillus and Clostridium there, the hardiest of bacterial structures, difficult to kill with heat or chemicals. Autoclave, 121 degrees C. So that's 21 degrees C higher than the boiling point of water. And the pressure, ridiculous. Okay? And that's basically what an autoclave is. It's just, like I said there last week, it's just kind of a big pressure cooker. And that's what it is. That's all it is. Huge pressure, huge heat, and in theory, it kills all the bugs inside of it. Okay. This is I included this here. I like this. The purpose of this spore, not reproduction at all. It's just survival. So when the bacteria realize there that, hey, I'm probably not going to make it, not the best time here to be around, so we're going to make a spore. This thing here is just there. It'll sit there for as long as need be until the conditions are right, and then it'll come back. So you see the problem here is that these spores can live for thousands and thousands and thousands of years, so that an entire 100,000 years, so basically an entire ecosystem from where this bacterium came from is now gone, and now there's a whole new ecosystem there with new animals, new fauna, new flora, and possibly this fauna and flora are not going to do too well against this bacteria that's been out of commission for 100,000 years. This is a scary thing. It's so unpredictable. The bacterial cell membrane, we're going to get into a little bit of the peptidoglycan. We're going to get into the membrane and the cell wall. Okay, just a little bit. Okay. The cell membrane here regulates what moves in and out of the cytoplasm. Okay, it's the mechanism there to bring things from the outside inside, or things from the inside to the outside. And there's a couple ways that things tend to move. There's diffusion. We've talked about this already, okay? Where things move from a high concentration to a low concentration. And just the whole idea there, if I take a little squirt of Ralph Lauren cologne or perfume and squirt it in the corner at the opposite side of a gymnasium. So I just spray it into the corner 30 meters away on the other side of the gymnasium, and within a couple of minutes, the smell should permeate the entire room, right? The high concentration is in the corner where you sprayed it. The low concentration is the air in the entire gymnasium, and things move from a high to a low concentration. It's called diffusion. Osmosis is the movement of water, and water only, across a membrane. And I just put a couple words in here. They're isotonic. Just means having the same osmotic pressure or concentration of fluids dissolve salts as your body. So it means isotonic means basically the same concentration as something else. Hypertonic means it has a higher osmotic pressure or concentration of fluids or salts as compared to your body. Okay? Hypo means lower. Okay, so you got isotonic, hypertonic, hypotonic. There's different kinds of, there's a whole bunch of membrane transport systems. we're only kind of looking at two, okay, and not really even looking at them. There's passive membrane transport, which is free. It's free money, okay. There are some things there that can move across. There are some things there that cannot. Like some things there that can't freely move across is proteins. They have to be packaged. They require ATP in order to move. There's another word there that I'm sure you guys have heard of. Okay, phagocytosis. Okay, that just means Pac-Man. That just means chomp chomp. Okay, there are white blood cells have the capability of phagocytosis. Chomp chomp, eating another cell. Okay, there are these things here. Okay, so great word there, phagocytosis. Okay, and I actually put chomp chomp. So there's a couple words there that you might see. Okay, the bacterial cell wall. It's a macromolecule, a polysaccharide. Saccharide is sugar. Okay, poly means many. Okay, so that's what a polysaccharide is, many sugars. Okay, macromolecule, it's huge. And just these repeating sugars there, and they kind of cross-link, okay, with short chains of amino acids. Okay, so it's a mixture there of sugars and protein, okay, and this is what we call peptidoglycan. Okay, tough outer coat prevents rupture, protects the cell wall, gives it its distinct shape, and is the starting point there for some antibiotics to get in and break it. So if you break this peptidoglycan, the cross-links there, now you've got a hole in the bacteria, and now you have a way to get in and destroy the bacteria. Okay, that's all we're looking for there. Nothing too crazy. And you'll see here, I included this. We want to go into a little bit of these cell walls because we're going to get into a little bit of gram staining. And as I said, this is better done in the lab. All right. So that's why we're just going to be taking a really, really, really flat stone and throwing it against really, really still water. And it's just going to skip. That's all we're doing. It's just skipping the surface, just so that you guys will have a little bit of awareness of this. Of course, please take a microbiology class and do this for yourself. It's a lot of fun. Going back to this slide though, cell walls and cytoplasmic membranes of a gram-positive bacteria. So this is what we call a gram-positive bacterial cell wall. Okay? And I'm going to go into the little differences here because it'll make a little bit more difference here. But the gram-positive bacterial cell wall has a thick peptidoglycan layer. So this is your peptidoglycan layer right in here. It has a nice thick one. Okay? Okay. The gram-negative bacterial cell wall, okay, has a thinner peptidoglycan layer, and it is separated there by 2. Okay, it's in the middle. So the gram-negative bacterial cell wall contains what these liposaccharides, lipofat, saccharide, sugar, phospholipids, and proteins in an outer membrane linked to a thin inner peptidoglycan layer. So there's your thin, and then you've got a cell wall on this side and another one on this side. Okay, this is for your mycobacteria. Not interested just yet. Okay, so the differences in staining, and I've got a movie here. We'll just play it and see what you all think. And just to let you know that gram staining, this is the action there that we, in taxonomy, in trying to name these bacteria, it's one of the first things there we do. And there are what we call gram-positive bacteria and gram-negative bacteria. And this is after you stain and do the whole process, this is what they look like. And it's one of the first things we got to do because in identifying these, that's how we determine their pathogenicity, their dangerousness. Let's have a look.
Grammarly can't teach you to dance, but it can help you make a great impression for free. As you type, Grammarly ensures you come across.
Grammarly. I was thinking about getting that there for the kids. How is it? I don't know. If anyone has any interesting advice about Grammarly, stick around after all. I'm interested. I'm looking at that. You see the commercial, so I guess it's got to be doing all right. But anyway, let's go.
Bacteria have cell walls made up of polysaccharides that give them strength and rigidity. This is important since bacteria often experience variations in osmotic pressure due to the different solutions they encounter.
And you see, you have a look here. Higher concentration on the outside, lower concentration on the inside. What's going on here? This is a higher concentration outside the cell. Here's an isotonic, okay, where it's the same on the inside and the outside. Hypertonic, hypo. You see? Get it. Good.
And it is their cell walls which prevent them from shrinking or swelling. As a reminder, osmosis is the process by which solvent molecules pass through a semi-permeable membrane from a less concentrated solution to a more concentrated one, equalizing the concentration on either side of the membrane. Nearly all bacterial cell walls have a peptide polysaccharide layer called peptidoglycan, also known as purine. Peptidoglycan is a polymer made up of sugars and amino acids which forms a kind of mesh. Bacteria can be classified based on their reaction to the gram stain, which identifies them as gram-positive or gram-negative, based on the chemical and physical properties of their cell walls. Gram-positive bacteria have a thick cell wall, which consists of up to around 30 layers of peptidoglycan. This cell wall surrounds a monoderm, which is a single plasma membrane. Gram-negative bacteria have a much thinner cell wall, consisting of a single layer of peptidoglycan. This layer of peptidoglycan is sandwiched between two lipid bilayer membranes called diderms. We can differ.
Now, just to get back into, you see here, the gram-positive there has a thick cell wall, okay, and there's your peptidoglycan in here, all right. You'll see there that for gram-negative, basically have two cell walls there, and in between there is your peptidoglycan layer. And if we go back, if I can go back, this is what we're talking about here. Here's your gram-positive, right? Okay, there's your cell wall with your peptidoglycan, and then here's your gram-negative there with your two cell walls sandwiched there with your thinner peptidoglycan layer in the middle. Okay. Now how do I get back to? Where did that go now? Interesting. Perfect. Let's do that again. Will that come up? That's what I wanted. Perfect. Sorry.
Two lipid bilayer membranes called diderms. We can differentiate between gram-positive and gram-negative bacteria by dyeing them with crystal violet and then washing them with a decolorizing solution. Then a powder stain is added, for example, safranin or fuchsine. Gram-positive bacteria will retain the crystal violet dye and remain purple, while the gram-negative bacteria will be stained pink. Note that...
Very important. So the gram-positive there Okay, after this whole process, and I'll show you. I have more slides there. It'll go into the the gram staining process, but the gram positive there, after the whole process, they will be purple. The gram negative after the whole process will be pink. Okay.
Gram-positive bacteria also pick up the pink color of the counterstain. However, this is not visible when they are dyed with the darker purple color of the crystal violet stain. The reason for these staining differences is due to differences in cell wall structure, which is the chief difference between gram-positive and gram-negative bacteria. The gram stain detects peptidoglycan, and since gram-positive bacteria have a thick multi-layered peptidoglycan layer, they retain the crystal violet dye. Gram-negative bacteria do not retain the dye for two reasons. They have an outer membrane getting in the way of the crystal violet, and they lack peptidoglycan to retain the stain. Although both gram-positive and gram-negative bacteria can be pathogenic, gram-negative bacteria are more resistant to antibodies because of their impenetrable cell wall.
I like that. The gram negative are more resistant. Okay.
Unfortunately, these bacteria also develop resistance more quickly. Not all bacteria can be reliably classified through gram staining. Acid-fast bacteria and gram-variable bacteria, for example, do not respond to gram staining.
Okay, so basically what we're saying here is that in identifying a bacteria, the first thing they do is they'll do gram staining. Okay, and okay, so that'll tell us if it's gram positive or gram negative. But we have to do a subsidiary test after this because you've got these ones, acid-fast bacteria and gram-variable bacteria, that can, what's the word I'm looking for, can hide, okay, and they can appear to be something that they're not, okay.
Acid-fast bacteria are bacteria whose cell walls retain stains particularly well. Although they aren't closely related to gram-positive bacteria, they can appear purple after the gram stain test. Gram-variable bacteria show a mix of pink and purple cells when stained. If you like this video, like and
Okay, and that was a good little introduction there. We'll just go on here. So the gram staining is a different stain procedure using in what happens here if it's gram positive there. Okay, you if you just look at these under the microscope, they just look clear. You really can't see them at all. So that's why we do staining, right? Because the light comes up there through, and actually we do a whole week on, I was actually going to include some microscopy on here, but I just thought I'd hold that. I don't want to confuse you guys too much. But we do spend a week there and talking about microscopes and these kinds of things. But that's why we stain these, to give them some kind of color so that we can see them, right? So the first thing we do is crystal violet. Okay, stains them like this, and then we treat them with iodine. And then there's usually a decoloring agent, alcohol, and then there's a counter stain, saffron. Okay, and generally that's I think they also use fuchsinin as well. Okay, and that gives you that, and that's how we tell. Gram negative is pink, gram positive will be purple. Here's another slide, the same banana. Okay, if you didn't like that or the video, say crystal violet, iodine, alcohol, and saffron. Okay, and that's how this one goes. And you see right here, alcohol wash there, decolorization, and then you use the saffron of fuchsin there, and that's how you tell. Okay. The acid-fast, a few species of bacteria do not bind simple stains readily. Okay. Mycobacterium in specific. Okay, acid-fast staining employs heating. So in addition to what we just did there, we'll have to heat these guys, okay? Heat the mixture, basic fuchsin and phenol, followed by counterstaining with methylene blue. So there's an extra step going on here for these acid-fast bacterium. As they have a high lipid content, a high fat content, and so decoloration by alcohol doesn't really affect them. Because lipid is a covalent bond, non-polar. Alcohol is very polar. Go back to your introductory chemistry, and so the alcohol really doesn't affect these guys at all. And the non-acid fast cells are decolorized, and then they're counterstained there with the blue. Okay, so the acid fast, there's an extra step here. You got to heat them up. Okay, and you'll see there the non-acid fast bacteria there. They show up like this. Okay, but with your Mycobacterium tuberculosis, okay, that's how we tell. Okay. Staining specific structures, they're the most important structure specific stain for bacteria involving visualization when bacteria have formed into endospores. These are that's it. Very difficult to kill. Important to understand in the context of a sterile processing environment. Remember, this is all being applied there to some type of a health environment setting, whether it's a dentist's office, or a hospital, whatever it is. Okay, and these are what autoclaves are used for, to get those spores. Okay, and just some different shots here. You know what I mean? You've got the spores in here. Okay, and you'll see the little spores in there. All right, and these are the ones there that someone here in a MDR, medical device reprocessing, has to be exactly aware of. These spores are the tough ones. Okay. That's bacteria there, kind of structurally, a little bit of history, these kinds of things and how they relate to us. And again, we're just throwing a flat stone against flat water. We're just skipping. And there's so much information. I encourage you guys to go out and do your own research and have fun with this stuff. Okay. We're kind of moving on there to bacterial nutrition, which we'll get more into there next week. Okay. And bacterial growth and these kinds of things here. But some bacteria, they live separately. They're loners, and others remain in colonies. Okay. And these colonies are large groups of bacteria. Okay. There are bacteria that live as parasites. And a parasite absorbs food material from other living hosts, from other living organisms. They're called their hosts. And a parasite, in most cases, their parasites really don't, they can abnormally affect a host, but generally it kind of defeats the purpose. Why would a parasite kill its host? You're killing its food supply, but some parasites do. Others digest and absorb food material from dead organisms. Okay, these bacteria are called saprophytes. Okay, just different there. Okay, and we've spoken about this there, and we'll go on to it there even more. Beneficial bacteria. Most bacteria are beneficial, helpful to humans. Like just the whole idea. If I know it's not, if a squirrel dies on a squirrel fell out of a tree or something there in your backyard, it's not going to, on a sunny day, it's not going to last too long. The bacteria will be all over that. That'll just, that's it. And it's just a way of cleaning the environment. Okay. Bacteria are decomposers that cause decay. They break things down. These decomposers are organisms that break down dead organisms and return nutrients back to the soil. It's the circle of life. Okay? And nothing beats the bacteria. Okay? Therefore, food and life-saving drugs. We already know this. Cheese, pickles, yogurts, vinegar, kimchi, sauerkraut, all these kinds of beer and wine, life-saving drugs, all these kinds of things, okay? And that's one of the things there, the bacteria, that's part of the world there that we're just starting to get our head wrapped around, this whole universe there that we can't see, and the ability there to genetically manipulate bacteria to eat oil spills. I think is a great thing, because I hate oil spills. They just kill millions of animals, and you wish it'd be gone there someday. So, yeah. By using methods of gene transplanting, bacteria have been encouraged to produce substances such as insulin. That's a good one. Okay, so we can have some fun and see what they do is they take the genes there that code for insulin out of an animal cell, Okay, through genetic recombination, add this, incorporate, and so the bacteria incorporates it into its plasmid DNA there, and now this bacterium is now producing insulin. Wonderful. I love it. But then you look on the inside now, you look on the flip side, now you have created a bacterial cell that has never been seen before in Earth's 4 billion year old history. What is this new bacterial cell capable of doing? You know, that's the whole thing. It's playing with evolution. We've all seen Jurassic Park. The human intestinal tract contains millions of good bugs. We've spoken about this there before. And again, there, be careful if you're taking antibiotics there unnecessarily. These antibiotics will wipe out all of these good bugs, and you need them. Many of these bacteria help the digestive process and help us extract nutrients and vitamins. Bacteria is also used in the tanning of leather, the curing of tobacco, and the production of food and feeding for cows. The harmful bacteria, this is what everyone's really interested in. Some bacteria are not beneficial to human. Okay, bacteria spoil food by secreting enzymes into the food, causing it to rot. We spoke about botulism. Okay, if that can of tomato sauce goes when you open it, throw it out. Along with enzymes, other substances produced by the bacteria are released into the food. Some of these substances are toxic and will kill you very quickly. Bacteria in large numbers can cause pollution. Okay, we've seen discoloration there of lakes. If sewage water is not properly treated, then it can cause damage there to the environment, drinking water, lakes, streams. We don't want to, ecosystems, they're balanced. There's this homeostasis kind of a thing here where there's all these checks and balances. And that's how evolution and ecology and wildlife and nature works. It's a homeostasis. You can't have too many wolves. You can't have too many deers. There's got to be a balance. And that's the whole thing with bacteria is if all of a sudden now there's a huge amount of bacteria that you've just dumped into, I don't know, Lake Simcoe. And you guys can go down to the beach. It's right beside the beach is the Barrie sewer plant, where all of everyone's dirty water goes right to the sewage plant, and you can see it there right by the downtown. You can go down the downtown Toronto. You can go anywhere and see it, and you just all this sewage water. And if you ever get a chance, go to the sewage plant nearest you, and you'll see that they use bacteria on a large scale to clean the water. It's quite amazing. It really is, and it's a good day for the kids. Actually, I might do that. All right. During respiration there, the bacteria reduce the dissolved oxygen content in the water supply. So if you're dumping all this bacteria into a lake or a stream or whatever, these bacteria in great numbers have the ability to augment that ecosystem in a negative way. And you drain the river or lake or whatever of its oxygen supply, fish are all going to die, and if the fish die, everything else is gone. The a pathogen, we've gone through this there. It's an organism that causes disease or some type of infection. Okay, some bacterial disease of humans are tuberculosis, tetanus, strep throat, and they're right here. Okay, see if I can blow that up a little bit. Okay, very good. And we're going to talk a little bit about controlling, but this is going to be a general theme throughout the course. Okay, chemicals, antibiotics, preventative practices, these kinds of things here. But we'll get into more of this there next week. Please remember there that bacteria will be continued there next week in conjunction with viruses. We'll do prions. We'll do a little bit of mushrooms as well. Okay. Antiseptic, disinfectant, and antibiotics are used to control pathogenic bacteria. An antibiotic is a chemical that can stop the growth of some bacteria, and we've talked about this there earlier there, that antibiotics there, they can attack the peptidoglycan cell wall, okay, or they can go after the ribosomes. And if you go after the ribosomes, then therefore bacteria can't produce protein. And if they can't produce protein, then they're effectively dead. They can't infect anybody or anything. Okay. Bacteria are able to produce types that are resistant to certain antibiotics. Okay, and you guys remember there that the gram-negative are able, they tend to have more of a resistance, okay, than gram-positive. And when this happens, new antibiotics must be developed. And that's the big problemo, okay? That's the big problem. We got to produce more antibiotics. But we're getting there. We'll get more into that there next week. Bacteria can be killed by pasteurization, okay? Louis Pasteur came up with this there, and it's bringing a liquid up to temperature 70 degrees. Okay, what a normal day is 21 degrees, a hot day is 33, 35 degrees. If you were in Bahrain or Saudi or in there like I was, a hot day was 53 degrees. Oh yeah, it's a good one. Okay, but you're bringing the food temperature up to 72, 70 degrees. Okay. There's your milk there, 72 degrees, and they heat it up to 70 and cool it. And in theory, okay, you're getting, I think it's 99% of all the bugs in there. You're not getting all the bugs because if you just leave your bag of milk in the fridge, it will go bad in about a week because there are bugs in there. They didn't get them all. Like, we could boil this milk, but then if you're boiling it, then you're going to completely denature all the proteins in the milk, and therefore it's not going to be good milk. So heat it up to 70, 72 degrees, and away you go. Okay? Canning. Okay? That's another way of food preservation. Okay? Chemical preservation. And they've been doing chemical preservation, salt curing for thousands of years. Okay, you bring in your fish there from the catch there today, and you leave your fish out on the beach. You put salt on it. It keeps the fish free of bugs. Dehydration, that is another way of food preservation. Steam, pressure cooking, sure. Radiation, okay, the gamma radiation there for your hot dogs. You know what I mean? I don't even know if you really need to keep a hot dog in the fridge. You could probably just keep your hot dogs on the shelf. Okay, what do we got now? There's botulism. We've spoken about that there, y'all. You guys can have a read of that. Okay, some bacteria can live anaerobically, and this is why I put this in here. Aerobic means needing oxygen. Anaerobic means living without oxygen. And that's what makes this botulism so bad, because if it gets into your food, they close the lid on the can, seal it permanently, right? And the next time you open it is at your house. Well, if the botulism bug was in there, it's just been having a happy old time. Okay, living in the dark, living in that sealed anaerobic environment. And then when you open it, there's the gas release that that was inside the can. That's where you throw it out. Two other anaerobic bacteria can cause venereal diseases. You've got gonorrhea and syphilis. Okay, both very bad if left untreated. Okay, gonorrhea causes sterility, and syphilis can result in death if left long enough and unchecked. A venereal disease is a contagious disease that a person gets there through sexual contact. Both of these diseases can be treated easily by antibiotics if detected early enough. Okay, so you see, the antibiotics is a great thing when needed. And the last slide here, I've given you guys some bacterial diseases in human. Have a look. Botulism we've talked about. Some of these here we haven't talked about, and I'll never talk about. But, if there's any in here that I've spoken about, then have a look. And maybe you might see them on a little test. There you go. But, ladies and gentlemen, and anyone else there, I hope you guys are doing all right. That's what we've got there. And that's today's class. And are there any questions? I'm still recording, and if you guys have any questions, let me know. And I'm going to stop recording. Actually, no, I'm not going to stop recording. I'm just going to recap what we're doing there for the next week or so. Okay, so this week here we did a little bit of introduction there on bacteria. Next week we will continue and finish up bacteria. Okay, then we're going to do a little bit of virus. Okay, because it's important stuff. Okay, with COVID and all this, we got to go into some viruses, and I got lots of good videos there, so please don't miss next week. Well, you can't because it's quiz 1, but we'll do viruses, we'll do prions. Prions, Google mad cow disease. It's a new one. And apply all this stuff there to your 10 identifiers of life. Are viruses alive? You know what I mean? And there's lots of good stuff here. We're still only touching the tip of the iceberg. We're only on week 3. And so I wish you guys all the best. If you guys have any questions, shoot me an email, shoot me a question. I'm right here right now. I hope you guys are doing all right. I'm going to stop recording now. But where we're at is next week we're going to finish the lecture there, and then after the lecture finishes, and then for both groups there, the quiz will open up at 12 o'clock, okay, next Saturday. And I think I'll leave it open for the week so that, because I want you guys to study week 4 before you do the quiz. Do not do the quiz before you have watched week 4 video, okay, and you understand it very well. And if you have any questions, shoot me an email. But the quiz will be open there for a week. I don't know if I'll leave it open for an hour or 2, but I will let you guys know. And that's that. The week after is Thanksgiving, so there is no class. And then week 6 will be the test 1. So there's no class on week 6, okay? The test there will be during, I'll give you guys more details on that, but your test will be in a couple weeks there, week 6. So hopefully there you guys are okay.